Good afternoon, ladies and gentlemen. Welcome to the first Taiwan Bridges keynote speech in Academia Sinica. We are honored to host today's session, which features insights from Dr. Roger Kornberg, the 2006 Nobel Laureate in Chemistry. Before we proceed, the president of Academia Sinica, Dr. James Liao, will deliver the opening remarks and formally welcome today's speaker. Please welcome President Liao.
Well, good afternoon, ladies and gentlemen. It's my great honor and pleasure to welcome you to this first lecture series in the Taiwan Bridges program held in Academia Sinica. Taiwan Bridges program was co-sponsored by International Peace Foundation, Academia Sinica, and National Taiwan University. And this is the inaugural lecture in Academia Sinica. And we are very pleased to have Dr. Roger Kornberg as our first speaker. And Roger Kornberg is not a stranger to Academia Sinica. He has been to Academia Sinica at least twice. The first time was 2009, I believe, as our Academia Sinica lecturer. And Roger Kornberg is a household name for people working in molecular biology. Roger and his father, Arthur, work out the most fundamental part of molecular biology, how DNA replicates itself and how DNA is transcribed to RNA, for which Roger won the 2006 Nobel Prize in Chemistry. And his father, Arthur, won the Nobel Prize in Physiology or Medicine about 40 years earlier. And both set the very foundation for how we can treat diseases today. So today, Dr. Kornberg is going to talk about something broader, where disease ends. I'm sure we'll learn a lot and enjoy his lecture. So let's welcome Dr. Roger Kornberg. Thank you.
Thank you, President Liao. Next, we would like to invite the Director of Institute of Biological Chemistry, Dr. Todd Lowry, to introduce today's keynote speaker. Please welcome Dr. Lowry.
So good afternoon. So it's a pleasure today to moderate this Taiwan Bridges lecture given by Professor Roger Kornberg from Stanford University. You didn't come here today to hear me talk, and so I'll be relatively brief. So as described by President Liao, Dr. Kornberg has had an extremely distinguished career in the field of nucleic acid biology, and of course won the Nobel Prize in Chemistry in 2006 for his work on the structure and mechanism of RNA polymerase II. Professor Kornberg's research exemplifies the power of fundamental, curiosity-driven science. By resolving the structures and mechanisms of the transcription machinery, his work has provided crucial insights that inform research areas across molecular biology, genetics, and medicine. These structural pictures do more than satisfy intellectual curiosity. They provide the mechanistic foundation upon which technologies are built. Today, we'll hear his continued achievements in this area, in particular, how his work relates to future pandemics. I want to express Academia Sinica's appreciation for Dr. Kornberg's making the time to visit with us, and also the International Peace Foundation for their support of this initiative, as well as our many partners who helped bring this lecture series to life. I also want to thank the Academia staff, predominantly those in the Department of International Affairs, for their hard work in making this lecture a series a success. So with that brief introduction, please join me in welcoming Professor Kornberg to give his lecture.
So thank you, President Liao. Thank you, Dr. Lowry. And thank you, Mr. Uwe Morowitz, for this opportunity to speak this afternoon. First, can I be heard well in the back? It is okay. Great, thank you. As you've heard, I've visited the Academia Sinica over the years and greatly admired the institution, and it's truly a pleasure to speak here again today. I've also spoken at many locations in Asia over the past several years as a guest of the International Peace Foundation under the direction, leadership of Mr. Uwe Morwitz. Always in the past, those lectures have been to lay audiences, to members of the general public. And in those lectures, I've taken the opportunity to explain what is basic science and to explain the significance of basic science. If I were to deliver such a lecture again now to another lay audience, I would speak of the emerging threat to science, a dire and a deeply concerning threat to science, and the threat to scientific truth everywhere in the world today. These threats have already slowed human progress, and they have profound sociological and, as you know, also political significance. They can't be fully addressed by us, all those gathered here today as scientists. They can only be effectively addressed by governments as agents of society. Sadly, in the United States, where the science of the past 75 years really began, the government today is leading in the opposite direction. Challenges to science and truth are rampant around the world, but the crisis in the United States matters most because it has been the engine of scientific growth. I doubt that many here, perhaps if anybody here, knows the entire history of post-war scientific growth. The United States emerged from World War II with extraordinary prosperity and belief in the future. The American Congress, in the most far-sighted act of its history, sought to put American wealth to work for humanity by the creation of grants programs. They enabled the National Science Foundation, NSF, and the National Institutes of Health, NIH, to grant unprecedented sums of money for research by any worthy investigator anywhere in the world. The NIH and NSF grant programs were based on two principles: individual investigator-initiated grants programs and peer review of their proposals. These two principles were both entirely new, they were transformative, and they were profoundly influential. Previously, funds for research were doled out in very small amounts by universities and always to the most senior scientists, which hindered the development of young scientists and especially preventing the pursuit of their ideas. This was particularly harmful because young scientists are responsible for almost all important new ideas. I have no idea why, but scientists are rarely original past the age of 40. I did my own most important work at the age of 25, not the work for which I was later awarded the Nobel Prize, but still my most important work. The NIH and NSF programs made awards all around the world for any good ideas, regardless of the age, the seniority, or the position of the applicant. Peer review then took the decision-making progress out of the hands of local university politics and entrusted the decisions instead to panels of leaders from everywhere in the various fields. The result was a truly merit-based allocation of funds on a scale sufficient for the rapid development of research. The previous hierarchical structure of academic departments everywhere in the world, but especially in the United States, was largely eliminated. It was emulation of the American model that improved the situation in the rest of the world, although hierarchical structures I find from my visits in the world still limit growth and productivity in many Asian countries and also some European universities. The NIH and NSF supported Nobel Prize-winning research everywhere in the world, in Europe, Asia, South America, as well as North America. Like any good thing, these programs have gradually become infected by politics, in the United States especially, and they were already in a state of decay before the current American administration added a crushing blow of not only ignorance of science, but also antagonism towards science. With that gloomy introduction, I'd like to turn to something joyful, and that is the science that we all love and all continue to pursue. As I mentioned at the outset, this is the first audience of scientists to whom I've spoken under the umbrella of the International Peace Foundation of Mr. Morowitz. And it is, of course, a superb and wonderful scientific audience to whom I would like to tell two scientific stories, if time permits. The first comes from industry, associated with concerns of a different kind that we could speak about in discussion later, but also real concerns. The second story will come from my own Stanford University laboratory. Both stories pertain to the so-called central dogma of biology, which is, I'm sure, well known to all of you, but it's a starting point for both of the stories I will tell today. Now, this, of course, relates to both the replication of DNA and thus heredity, but especially in what I will have to speak with you about today, the expression of genetic information through, as you know, transcription from DNA into RNA, translation into protein. The first story I'll tell you relates to a therapeutic intervention at the level of RNA, and the second concerns the very first step in the transcription from DNA to RNA, one that involves a profound change in structure of the chromosome, not seriously investigated until what I will have to tell you later in this lecture. Now, the first story that I will tell relates to the title of this lecture, which is a half-serious title, The End of Disease, but with a question mark. So it's a play on what some of you may know, an influential discourse due to a man named Francis Fukuyama, must have been about 25, 30 years ago, on the subject which he called the end of history. And it was the optimistic view at the time that democracy was progressing everywhere in the world, and the political history would come to a glorious conclusion. Of course, that proved to be a most unfortunate prognostication, and we're still in trouble everywhere in the world today. The end of disease, which, with a question mark, which I entitled this lecture, is probably more likely to achieve a successful conclusion, and I'll comment further in a moment. But to continue, as I say, I'll speak in the first of these two scientific stories about an intervention in the process of gene expression at the level of RNA, and especially as an example, it relates to future pandemic preparedness, but as I'll tell you, the application is far broader. And it relates in the case of the problem of the danger of pandemic disease to not only SARS-CoV-2, which and the whole family of coronavirus is well known to you, but even more so, influenza, more likely to be the source of the next global pandemic. This slide simply illustrates there have been 4 pandemic or epidemic events in the development of influenza A only in the past century alone. And it is a source of very great concern to people in the public health sector today because of the still developing and spreading H5N1 influenza, which today is found mostly in animals, but with a single mutation could jump to humans, where from the cases where it has occurred already has resulted in a very high fatality rate, 56%, a very frightening prospect. Now, the solutions to the problem of respiratory viral disease and pandemic spread in particular, up to this point, have taken two forms. Conventional drugs such as small molecules like Tamiflu, and also conventional vaccines such as that available to us in general today. The problem is these take anywhere from months in the case of vaccines, especially RNA vaccines, to years to develop in the case of small molecules. So a response to any new virus cannot happen rapidly enough to prevent pandemic spread. I'll describe briefly an approach which has been developed in a small startup company in Israel whose research I help to direct, that employs small interfering RNA. Some of you may know about the importance of these molecules. They're only about 20 some odd nucleotide base pairs in length. When they encounter an RNA molecule in a cell with the same sequence, the same genetic code, the same sequence of nucleotides, they bring about the destruction of that RNA molecule. So an siRNA, a small interfering RNA with carrying the sequence of a viral gene, will destroy the viral genome and terminate an infection. The important further point, the activity of small interfering RNA discovered by now almost 3 decades ago by my colleague at Stanford, Andy Fire, is a natural mechanism. So the capability of what I have just described, namely siRNA-mediated destruction of RNA, is present in every cell in the body, and of equal significance, an RNA-based drug, a natural mechanism that benefits from the exquisite specificity of nucleic acids, something which was derived by evolution, and so unlike almost all other drugs, no possibility of off-target effects, no likelihood of toxicity, and very high specificity. Now, the challenge with the use of small interfering RNAs is that of delivery to cells, and a case that concerns us here, human cells. In contrast with a typical small molecule drug, such as that shown here, of say 300, 400, 500 molecular weight, a small interfering RNA of 25 nucleic acid pairs is both 1,000 times larger, 100 times larger in mass, and also possesses, if in the case of a 25 residue double-stranded nucleic acid, 50 negative, fixed negative charges, phosphate charges, which make penetration of a cell, passage across the cell membrane, a virtual impossibility. Now, at the small organization in Israel that I mentioned, this problem was solved by the use of a specially designed steroid derivative, really a derivative of cholesterol, which, if attached to the two ends of the small interfering RNA, would enable its rapid penetration across the lipid bilayer membrane of any cell, whether human, bacterial, or otherwise. The way in which this was designed results in the rapid release of those steroid derivatives from the ends of the small interfering RNA following entry, as a result of which the small interfering RNA is trapped inside the cell. This has an additional benefit, as I'll shortly describe. Now, this simply illustrates the efficiency of entry. So a red dye labeled small interfering RNA exposed to cells in culture, which were then harvested and examined for internalization of the siRNA, gave the results shown here. In the case of the siRNA alone, as expected, without the attachment of the cholesterol derivatives, no detectable penetration. The small interfering RNA, dye labeled red with cholesterol attached, resulted in penetration of every cell to a significant degree in a short space of time. Indeed, over the course of about 30 minutes, every cell penetrated to the point of equilibrium. Finally, the effectiveness for therapeutic, both preventative and therapeutic purposes, was shown by experiments performed in a non-human primate whose respiratory system is closely similar to the human respiratory system. So in this case, a small interfering RNA with the sequence of a highly conserved region of the RNA-dependent RNA polymerase of SARS-CoV-2 was introduced by inhalation by African green monkeys. In this particular experiment, the monkeys were previously infected with SARS-CoV-2, and what you see in the example here where a therapeutic dose of siRNA was introduced, the blue columns, within a couple of days, there was a near complete elimination of the virus. This is a log scale of the number of copies of virus per milliliter in a nasal swab. By the end of day 6, 99.9% of the virus had been eliminated. Now, we know from the one other example of any effective antiviral drug in all of pharmacy, which is sofosbuvir for hepatitis C virus, that level of reduction of viral load is sufficient for a cure. The only other therapeutic that is widely used today and effective for SARS-CoV-2 is Paxlovid from Pfizer. It results in only a 10-fold reduction in viral load, and once the drug is withdrawn, the virus rebounds. It doesn't result in a cure, but it somewhat alleviates the symptoms of the disease. So just to conclude in this regard and contrast small interfering RNAs as a solution to the problem of future pandemics with the RNA vaccines, which are currently in use, whereas some months are required to develop a vaccine for a new virus using the RNA vaccine approach, the application to a new virus is virtually, of an siRNA to a new virus, is virtually instantaneous, because as soon as the sequence, which can take only hours to determine, has been established, then the small interfering RNA is made automatically on a machine. It can be made in any amount, can be made at very, very low cost. So within hours to days after a new virus is detected, already the drug can be widely distributed. It's important also to note that every new virus requires a new RNA vaccine, whereas a small interfering RNA directed against a conserved region of a viral genome will be effective against all forms, including any variant. So in the example I just showed you, the small interfering RNA directed against SARS-CoV-2 directed against a portion of the gene for the RNA-dependent RNA polymerase of SARS-CoV-2. That one single drug is effective against all 6,000 isolates that have been sequenced so far of SARS-CoV-2. Most importantly, as we know, the RNA vaccine must be maintained at ultra-low temperature to preserve its activity, whereas the siRNA is stable indefinitely at room temperature. The RNA vaccine must, of course, be administered by a medical professional, but can be self-administered by inhalation. It's noteworthy that immunity elicited by an RNA vaccine takes some weeks, whereas inhalation of a small interfering RNA will have a virtually instantaneous effect, preventing and also, if used as a therapeutic, curing the disease. Vaccines are, in the best cases, effective against most but not all individuals. A small interfering RNA will be effective for every individual to whom it is applied. And lastly, as I've mentioned, the RNA, once it enters a cell of the respiratory epithelium, is trapped in that cell, so it is a topical treatment. It does not penetrate the system, it doesn't enter the circulation. So that enhances the safety consideration. Because it is highly effective and long-lasting in its effect, but not an actual vaccine, there's even hope that in the United States today, where there is extraordinarily an individual in charge who doesn't believe in the power of vaccination, maybe this will take hold because it isn't really a vaccine. But that remains to be seen. Now, this was the application for future pandemic preparedness was merely an example. The application is much broader. So what I have just described is obviously immediately applicable to all respiratory viruses with the same facility. There are 23 common human respiratory viruses. There's only one drug against influenza, Tamiflu, developed in the past, and it has unfortunately ceased to be very effective because of resistance that has arisen in all recent variants of influenza virus. And then there is Paxlovid. But as I say, 23 common human respiratory viruses. It would be possible to immediately develop a small interfering RNA effective by inhalation against every one of those viruses. I've learned that some 75% of all doctor visits in the world are for people complaining of respiratory symptoms, which is hardly surprising because, after all, the mode of transmission is so rapid and effective. They are so infectious. It would be possible in this way to eliminate all viral respiratory disease. But that's just the beginning. We have actually today a study going on at Stanford University with a leading immunologist and pediatric oncologist, Dr. Michelle Mongi, who has identified a target for the treatment, indeed the possible cure of all brain cancers, of gliomas, one of the most deadly of all cancers. And she believes, and I as well, that there's a reasonable likelihood that a small interfering RNA directed against that target will provide the first effective relief from the scourge of brain cancer. And then with a colleague at Mass General Hospital and Harvard Medical School, Dr. Rudy Tanzi, we are applying a small interfering RNA that he believes will bring about arrest of Alzheimer's disease. So there are many potential applications. And it is really one of the reasons why I applied the whimsical title to this lecture, End of Disease with a Question Mark. Because by now there are many modalities. I mean, the small molecule approach, the classical approach to pharmacy and the treatment of disease is today supercharged by what many of you may know, the possibility of rapid development of small molecules using DNA-encoded libraries for their identification, and also the application of computation, including artificial intelligence, to speed the development of effective small molecules. Then I've told you about small interfering RNAs. I haven't mentioned, but the same approach can be used to deliver peptide therapeutics across the limiting membrane of human and other cells. It's believed that peptides will eventually be the most effective way of attacking some 80% of potential drug targets that are not approachable in any other way. And then, of course, all the wonderful immunological approaches that you're doubtless aware of: monoclonal antibodies, checkpoint inhibitors, CAR T, and then, as I've already told you, RNA vaccines. So I actually think that most disease will eventually be eliminated. My wife opposed my writing the title without the question mark. She's a fine scientist who doesn't agree that the prospects are so great, so I've done so. But I am myself bullish about the prospect. Now, in the remaining time, I'll tell you a scientific story that comes from my own laboratory that also relates to the possibility of intervention in the process of gene expression. Only what I'll tell you today is only about discovery. It's only about identifying the very first step in what will eventually lead to transcription of a gene and the production of RNA. And it may one day be another point of intervention, but the interest for me, and I'm sure for most of you, will be actually discovering the basis, more so than the eventual application to disease. Of course, the research that I tell you about is the result of work of very many investigators, wonderful students and postdocs who have been my colleagues over the years, and much of the work is published. The most recent work, which is what I will especially describe, is unpublished, and it is due to those listed in the slide here: Yali Lorch, my wife and closest collaborator, postdoctoral fellows Shigeki Nagai, and former postdoctoral fellow, but now longtime colleague, Pierre-Jean Matai. And then former student, I don't know why the pointer isn't working so well now, former student, now a faculty member of our department, Andrew Beal, and then longtime collaborators, Maya Ezubel and Donghua Chen. The problem is posed by the picture that is shown in the next slide, and it's one that is familiar to many of you. And a picture like this, a light microscope picture of a cell during most of the cell cycle at the top, and then a cell in the process of division at the bottom, familiar, I'm sure, to almost everyone in this audience. Pictures like this, taken through light microscopes for well over 100 years, show, as I say, a cell during the majority of the cell cycle within which the nucleus resides with a limiting membrane, and then in the center of the nucleus, the so-called nucleolus, where ribosomes for protein synthesis are formed. A cell in the process of division looks altogether different. The nuclear membrane has dissolved, and the chromosomal material distributed through the nucleoplasm, throughout the nucleus, and therefore not visible in the light microscope, of a cell during most of its life, most of the cell cycle, the chromosomal material now condensed in the form of visible, discrete chromosomes, two groups moving to opposite poles of what will become the daughters of the cell division and following cleavage across the center of this image. The next slide simply shows a nicer picture of mitotic chromosomes, because they are really the main point and the focus of what I will have to tell you today, and in the structure of the chromosomal material within these entities, the main outcome of what I will describe to you today. These are extraordinarily large. They're almost the size of the cell itself. and it's hard to believe that the detailed structural organization of such a massive cellular organelle could be deduced from straightforward investigation, but that is what I will tell you today. The next slide shows an electron microscope picture of a cell nucleus, which reveals some important additional detail. So only about 20% of the chromosomal material of those condensed chromosomes in mitosis decondense for the purpose of gene expression during the majority of the cell cycle. The material, that 20% that undergoes decondensation, is called euchromatin, and it contains all the genes that are active in transcription and is distributed throughout the interior of the cell nucleus. The 80% that remains condensed as in the chromosomes during cell division is termed heterochromatin, and it contains all the genes that are inactive in the particular type of cell, of course varying according to the cell type and the requirements for gene expression. So the process of conversion from heterochromatin decondensation to form euchromatin is the first step in turning on gene activity, the first step in transcription. It is the step where selectivity, the determination of which genes will be transcribed, first occurs. What I will tell you today is about the studies that we have done only very recently that shed light on the basis for that very first step, the decondensation of heterochromatin to euchromatin, and the structure of the chromosomal material in these two states. The story that I will tell you actually began 50 years ago with my discovery of the basic unit, the fundamental particle of the chromosome known as the nucleosome. The nucleosome idea, which I put forward at that time, summarized here, the nucleosome as an assembly of 8 histone protein molecules with 200 nucleotide pairs of DNA. Some 20 years after my proposal of the nucleosome idea, the atomic structure of the nucleosome was determined by Luger, Richmond, and their colleagues. And 2 views are shown here. What it revealed, as I had surmised and proposed at the time, the DNA wrapped around the set of 8 histone molecules. So here you see the DNA double helix in green and orange, wrapped, in fact, in 1 3/4 turns around the set of 8 histone molecules at the center of the nucleosome, with the 4 different histone types, 2 of each, in blue, green, red, and orange. The so-called core particle of the nucleosome, consists of what you see, a globular core, but also about 1/4 of the mass of every histone molecule protruding into solution from the nucleosome without any defined structure, in contrast with the globular core. These seemingly unimportant straggling tails turn out to be the most significant for the formation of the structure of chromosomes, as I will now explain. These tails, shown here in regard to their amino acid sequence, are, in the first place, highly positively charged. In fact, the histone proteins are among the most highly positively charged proteins in nature, but none of that positive charge resides in the globular core. All of it is located in these flexible, unstructured tails. And the charged residues, arginines, and lysine residues indicated in red and blue in these amino acid sequences of the tails. Remarkably, removal of the tails in their entirety has no effect on the structure of the central core particle. Rather, the DNA is bound to the histone molecules in the core particle entirely through weak interactions, which is a point of very great importance and fundamental to chromosome function and transcription that I won't have time to talk about today, because I especially want to explain to you how understanding the role of the tails has revealed the fundamental basis of chromosome structure. So I'll continue in that regard. Now, the question, quite obviously, from the images that I already showed you of mitotic chromosomes, is the problem of understanding the extraordinary contraction of length and condensation of the chromosomal material and the selective decondensation that occurs in the course of the cell cycle for the purpose of gene expression. There is approximately 10,000-fold contraction in length of the DNA in the mitotic chromosome and in heterochromatin. And it is widely believed, in fact, anyone you ask anywhere in the world today will tell you, well, that could only be achieved in a hierarchical manner, such as illustrated on this slide, namely, first coiling the DNA around the nucleosome, which achieves about a factor of 5 contraction of length, and then coiling the chain of nucleosomes, and then coiling the chain of chain, and so on and so forth, through approximately, say, 5 levels of hierarchy with a 5-fold gain of contraction at every stage, and when all multiplied together, 5 times 5 times 5 times 5, finally achieving a 10,000-fold contraction of length. What I will tell you now is that that idea is completely wrong. Even though widely assumed, and even though much evidence has been published to support the idea, it is flatly wrong. And the solution to the problem is so beautiful and so far simpler. So contraction of length is achieved through loop formation, and that is through the action of molecules called enzymes, called condensins, that are actually conserved from bacteria to humans. Now, the idea of loop formation, is not new. The formation of loops has been known for over 100 years from light microscope pictures of, for example, lamprey chromosomes in amphibians, in the meiotic stage of amphibians development, which show loops of chromosomal material radiating from a central axis. But loop formation only achieves contraction of length and no condensation. The evidence that, in fact, those processes occur by entirely different mechanisms was obtained by Earnshaw and his colleagues many years ago, showing that if condensins are destroyed during the process of mitosis, no chromosomes are formed, but the chromosomal material nevertheless condenses in a single globular mass. So loop formation with contraction of length and condensation, which reduces volume, are entirely separate processes. Loop formation catalyzed by condensins. And now I will tell you what is the basis of chromosome condensation and decondensation when it occurs in interphase of the cell cycle. The problem of investigating the organization of chromosomal material in the condensed state, it is not accessible by ordinary structural technology or methods available today. So it can't be investigated by X-ray diffraction because there's no long-range order, so no periodicity that could be detected. It's been tried and it's failed. And it can't be investigated, at least until recently, as we have done, by electron microscopy, because the density of the chromosomal material in the condensed state is so great that it prevents transmission of an electron microscope beam. So about 15 years ago, I decided we would take a different approach. We would investigate the forces responsible for condensation. And this we did by adhering chromosomes to a glass cover slip that could be viewed by differential interference contrast optics, installed in a perfusion chamber so we could flow solution of various composition and expose the chromosomes to different conditions, as I say, to try and understand the forces responsible for condensation. The next slide simply shows an example where a mitotic chromosome, in this case from a human HeLa cell on the left, was exposed to very low ionic strength conditions. So with the removal of both mono and divalent cations and only a small amount of buffer left to maintain the pH, and here it is expanded some 10-fold. Now, such swelling of chromosomes in the absence of counterion is well known. It had been observed before. What is different is we undertook to analyze it on a quantitative basis, and through measurements on hundreds of thousands of chromosomes to obtain precise information so as to finally understand the basis of the process. So this shows, just illustrates the feasibility of the approach. It shows several cycles of swelling, as I just described, and then recondensation by adding back a multivalent cation, the naturally occurring chromosome-associated cation spermidine, which carries 4 positive charges. And what you see is recondensation, and the process could be repeated, swelling by removing the spermidine and leaving behind only a small amount of buffer to control the pH, and then recondensation repeatedly. The process is evidently reproducible. The morphology of the chromosome is retained, so it is a non-destructive process and appropriate for analysis of the basis of the chemical basis of the process. Now, then, as I have mentioned, we had the opportunity of acquiring hundreds of thousands of images, and my colleague Andrew Beal, who I mentioned, wrote literally thousands of lines of code for the automatic analysis of these hundreds of thousands of images to correct, first of all, for motion, for variation in lamp intensity, for other fluctuations, and then to be able to properly evaluate the very diffuse images in the decondensed state. On the basis of what I have just described, we could arrive at the picture that is shown here, and this is the key experimental evidence whose significance I will shortly describe. So each of the data points here, each of the data points represents an average from hundreds of chromosomes where recondensation from the decondensed state was brought about by the addition of naturally occurring amino compounds bearing 1, 2, 3, or 4 positive charges. The interpretation of this information was made possible by a whole generation of physical chemistry done in the analysis of what are called ionic hydrogels. So physical chemists prepared, experimentally measured the behavior, and then developed theory to fully explain the properties of ionic hydrogels over a period of 50 years. The theory which they developed, which I will not describe in detail, is simply summarized here, and the essence of it is at the top. And that is that swelling of an ionic hydrogel is described by the simple equation for the osmotic pressure that you see here at the top. There are two terms for swelling. One, the mixing term, which refers to the natural tendency of any material to dissolve in solution and so expand. And then a term that refers, a so-called Donen term that refers to the entry of ions to neutralize the charge, accompanied by water to maintain their concentration, also drives swelling. And finally, the elasticity of the ionic hydrogel, which opposes those two processes, which at equilibrium, when they are in balance, results in zero of the osmotic pressure. Now, what physical chemists developed were equations showing in closed form of the behavior of these three terms, the mixing term, the elastic, and the ionic term. The ionic term, in our case, actually includes 9 additional components because of the complexity of our solution and the chromosomal material. The next slide simply illustrates the result. Our experimental data, when plotted in an appropriate fashion, correspond precisely to the predictions from that physicochemical theory. And on this basis, we could conclude that chromosomal material is an ionic hydrogel. Now, let me just explain with a diagram what exactly that means. So a polymer in solution may, through affinity of its component parts for one another, condense under appropriate solution conditions to a diffuse globule. But if the polymer is capable of internal non-covalent cross-linking, it will form a hydrogel. And if those cross-links occur between positive and negative charges, then a so-called ionic hydrogel. That is the nature of the chromosomal material, where the positive charges, of course, derive from the histone tails, and the negative charges are the phosphates of DNA. Now, what the physical theory predicts and what physical chemists demonstrated is a remarkable property of an ionic hydrogel, that under certain conditions it will undergo total collapse to an essentially solid state. It will undergo what is demonstrably a first-order phase transition, called a volume phase transition, because it corresponds to a reduction in volume. This is a physical phenomenon closely similar to a liquid-gas phase transition, perfectly analogous to boiling or condensing vapor, water vapor, or what have you. So what I showed you in the previous slide were, in fact, illustrations of a volume phase transition of the chromosomal material. And indeed, the chromosomal material following such collapse is in its most condensed state. I think it's important to emphasize for many in the audience who may be aware of the recent, often misunderstood so-called liquid-liquid phase transitions. There's a lot of recent literature on the subject. It's important to distinguish between a volume phase transition, such as I've just described, and this recent enthusiasm, often misguided, for what are called liquid-liquid phase transitions. What that refers to is the aggregation of low-complexity proteins that form gels. They undergo a sol-gel transition, such as I showed you at the top of the previous slide. A volume phase transition, by contrast, occurs between two thermodynamic states, from a gel to a solid state, analogous to the gas-liquid phase transition, like, as I've told you, water vapor condensation. So from this, we understand at first level the basis of mitotic chromosome structure. It begins with a linear configuration of loops formed by condensins, and then collapse of the material through a volume phase transition, collapse of a classical ionic hydrogel. Now, we know from additional analysis of those profiles that I showed, they exhibit what is referred to as hysteresis, which is evidence of disorder of the material in the collapsed state, and that we could demonstrate directly by electron microscopy. So what we have done is to swell only allow the swelling of mitotic chromosomes to a modest extent to allow passage of an electron beam, swelling to an extent of only a few percent, but sufficient at the edges for passage of the beam. And now, through what is called cryo-electron microscopy, familiar doubtless to many of you, and in particular cryo-electron tomography, we could observe, as they occur in the chromosome, nucleosomes. You see the double-layer disc so the result of the wrapping of two coils of DNA around the central histone octamer. And because the DNA that passes between those nucleosomes in the chain of a chromatin fiber could be seen and traced, we could actually follow the trajectory of individual chromatin fibers in the condensed state of the mitotic chromosome. What is noteworthy, of course, is the absence of any higher order structure. No coiling of the chromatin fiber, even in the most condensed state. So these chromatin fibers, of which many could be analyzed by this approach, never revealed what was thought unavoidable and believed to be the basis of chromosome condensation. So here's an image in which, through cryo-electron tomography, the locations of 520 nucleosomes organized in 18 different fibers, color-coded accordingly, with a density, a very high density, even when slightly expanded, of 10% weight, volume by volume, close to the density of the fully condensed chromosome could be observed. Finally, to conclude then, the unstructured histone tails with their high level of positive charge, through interaction with the negative charge of the DNA, give rise to the fundamental basis of the chromosomal material as an ionic hydrogel, which through a volume phase transition undergoes collapse to the most condensed state. The answer to the question about higher order structure, there is none. The chromosomal material is essentially random. In fact, it may be described as a two-state system, either expanded or collapsed, with expansion brought about by modification of post-translational modification of the lysine residues of the histone tails by acetylation, which cancels their charge, then eliminates the cross-links that are responsible for hydrogel formation and allows decondensation for the purpose of transcription. So it is a two-state system which can exist in essentially random organization in both states and passes between the euchromatin and heterochromatin states in the manner that I've described, which represents the fundamental basis for gene activation in euchromatin and repression, of course, in the heterochromatic state. The extraordinary outcome of this, it answers what was for me, it solves for me what was the paradox of the mitotic chromosome, whose images I showed you in the light microscope at the beginning. I puzzled for years, how could a heterogeneous chromatin fiber with different regulatory proteins and other components all along according to the sequence of the DNA, which varies along the entire length, condense to a well-ordered three-dimensional state. And the solution to that paradox of order arising from heterogeneity, the condensed state is formed by random collapse. In fact, at the end of the day, we might have anticipated what I have told you is the conclusion of this work. Naturally, nature has no use for organization of euchromatin, the expanded version, and has no need of organization at the level of a defined structure in the condensed state, simply expansion of the chromatin fiber, which follows on a more or less random trajectory in the euchromatic state, and collapse for the purpose of chromosome condensation and mitosis in a random configuration of the material in the condensed state. With that, I thank you for your attention.